So why is the statement antibiotics have created drug resistance in MRSA inaccurate? Remember, the drug resistance mutation has to be present prior to the antibiotic being present, not after. If there is no resistance mutation present on exposure to the antibiotics do what they're supposed to do, kill the bacteria. So the antibiotic can't trigger the drug resistance. What it can do, however, is select out individuals who have the drug resistance by killing off all of those that lack the drug resistance. That preserves those that do have the drug resistance. They are ones that thrive and survive and reproduce. So the antibiotics don't create the drug resistance. What they do is select for those individuals that have spontaneously developed the drug resistance as a consequence of mutation. Mutation comes first, the selective pressure comes second. That's always the case in natural selection and evolution. So let's move onwards. We've got a few more slides to get through, but we're getting close to the end. So what we're going to look at now is some of the evidence that Darwin had available to him that he could use as evidence for evolution. And we're also going to look at some of the modern evidence for evolution. So one of the pieces of evidence for evolution that Darwin would have been familiar with is the notion of comparative anatomy and the idea of homologous structures. So there are two ideas that you're going to have to become familiar with that we'll come back and address in a little bit more detail in another chapter again. The two ideas are homology. Let me bring it down again. So homology the other one is analogy. So homology is similar characteristics or similar traits because of common descent. Analogy is similar traits because solution to the same problem. And I'll show you an example of that a little bit later here. And what's important here is no common descent. So these are two ideas that you are going to see reoccurring many, many times over the course of the semester. Multiple times in the next few chapters, but then we'll come back and we'll refer to some of these again as we move through the semester. So homology, when we talk about homologous structures, what we're talking about are structures that are similar on a number of closely related species because they're all descended from a single common ancestor. So here's one that you might have been familiar with previously, but these are all forelimbs of mammals. And so they're all closely related because they share a descent from a common mammalian ancestor. And so when we look at the forelimb, what we realize is that these forelimbs, and in fact all limbs in general, whether they be forelimbs or hind limbs, have a similar structure, which was first recognized by a paleontologist named Richard Owen, who was a big deal in paleontology in the mid-19th century, so around the time of Darwin and a little bit later. And Owen was sent, because he was such a big deal in the world of paleontology, was sent a lot of fossils. And as he looked at the fossils and compared them with living organisms, he came to realize that limbs have a basic structure. And so looking at my arm, hopefully you can see this in the well, let me do it on the screen. Make sure Pen's working. So Owen's basic layout of a limb was one big bone, two big bones, cluster of little bones, digits. And when you think about your arm or your leg, you'll realize that that's the basic makeup of your arm or your leg. Now, it's the same basic set of bones. They're not exactly the same because they've been modified for different purposes, but they are the same set of bones. So you actually show a little bit of this idea of homologous structure between arms and legs. But if we compare forearms or forelimbs, so our human arm, with our cat, we see again, one big bone, two big bones, cluster of little bones, digits. So the cats are actually walking on their toes. And when you stop and think for a minute about how you contact the ground when you're running, you'll quickly realize that if you're running quickly, you're running on your toes. You're not running on the flat of your foot. So this clearly indicates to us that cats are going to run fast. That's because they're hunters, they're chasing prey. And so their leg has been adapted to that. A whale has the same form, one big bone, two big bones, cluster of little bones, digits. But now a whale is trying to move this limb in a very viscous medium, water, much more viscous, much denser than air. So there's going to be a lot of resistance to that movement. And so consequently, the leverage required to move the limb is going to be quite great. And you know that hopefully that with the forelimb of a whale, it moves it backwards and forwards in order to be able to move slowly through the water. If it wants to move quickly, it's going to tuck its flippers in and it's going to use its tail flukes. Now, those have a different origin. They're not from the hind limbs. Whales have lost their hind limbs, with the exception of some small bones. But again, that's an example of evolution at work and natural selection and adaptation. But the whale's forelimbs or flippers are going to need to be able to resist movement in the water as they are swept forwards, but then lock in place as an oar-like arrangement in order to be able to drive the whale forwards as it moves the flipper backwards. So the extreme flexibility of the flipper to allow it to be moved forwards is because of all of these tiny little digit bones, the phalanges. The leverage to move it against water and to provide the power to move it against water is created by shortening the arm bones and making them very thick and creating a very rigid structure that attaches this to the shoulder to create a very rigid hinging lever that allows it to move the flipper against the extreme density and viscosity of the water. The bat has a different problem. It's wanting to move its wing against the very non-fluid, non-viscous air. Now, air is a fluid, but it's not terribly viscous. So the bat's wing again has one big bone, two big bones, cluster of little bones, digits. And this time it wants a lightweight, broad surface area to move against the air so that it can move the maximum amount of air to allow it to fly and support its movement through the air as it's flying. So it does that by elongating the bones, making them very long and very thin in the upper arm bone, because it's not dealing with a viscous medium. And now it's got elongated digits, but these elongated digits are long and thin in order to support this skin-like membrane that creates the wing surface area. And this is adapted and modified to move large volumes of air so that it can fly. Notice it's the same basic structure, one big bone, two big bones, cluster of little bones, digits, because this is the basic structure that was inherited from the mammalian ancestor. But each of these limbs has been modified. There's the descent with modification, along with the unity, to be used for a particular purpose, an environment or a function that is unique to the life of these animals. And it also reflects the fact that it's all about tinkering when it comes to adaptation, because evolution can only work with an existing structure. You're not going to see a whole lot of circumstance where you're going to see a brand new structure develop using a clean sheet of paper design. It's all going to be about modifying and adapting an existing structure to a new use, and that's exactly what you see here. So evolution and natural selection tinker with existing structures to see what works and what doesn't work under new circumstances. And so the human arm, the cat's leg, the whale's flipper, and the bat's wing are all representative of tinkering experiments that have led to structures that work for the organisms that possess them. But they are tinkered, modified structures that are descended from a common limb structure that had one big bone, two big bones, a cluster of little bones, and some digits. And when we talk about the origin of terrestrial tetrapods and terrestrial animals towards the very end of the semester, we'll come back and revisit this idea, because there's another story to tell there. Then another piece of evidence that Darwin may have resorted to, because this discipline was in the process of developing, and the individual who was most noted for developing this was a contemporary of Darwin's, a German biologist by the name of Ernst Haeckel. Unfortunately, Haeckel has been misunderstood and his diagrams have been misinterpreted. But what Haeckel was showing was that vertebrates during their embryological development, so vertebrates means most of the animals that we are familiar with, and particularly mammals, but we could look at other animals that possess a vertebral column, belong to a group of organisms known as chordates. And chordates have a common set of features that you can see listed here. Pharyngeal slits or gill pouches, a notochord, which is the precursor to the vertebral column, a hollow dorsal nerve cord that eventually gets encapsulated within the vertebral column, and a postanal tail, which a lot of animals have, which we had at one point for about 3 days, but then got reabsorbed because we don't need the tail. So if you were a fish, the gills would develop into, or the gill pouches would develop into gills for gas exchange in water. We have the pharyngeal slits during one of our embryological stages, but they eventually develop into eustachian tubes that connect our ears to our throat so that we can adjust the pressure in the inner ear to match that of the throat so that our eardrums don't burst. Some of the bones that support or would have supported the gills, what are known as gill arches, have actually evolved into the two of the 3 little bones that connect the eardrum to the inner ear. They were once part of the jaw structure, but they're now in the inner ear, and we'll talk about that when we talk about the evolution of animals later in the semester. I've talked about the loss of the postanal tail, but some of our ancestors do still have tails. So remember, we belong to the apes. We belong to the great apes that are a group, most of whom have lost the tail, but one of the great apes, or sometimes referred to as a lesser ape because it does have a tail, is the siamangs, the gibbons. They do have a tail that they can use for working in trees, which is the reason why they're referred to as lesser apes. But then when you look at monkeys, which are primates, and we are primates as well, they have the postanal tail, but our common ancestor with them probably had a postanal tail, but as apes evolved from the common ancestor with monkeys. We dispensed with the tail because apes spend more time on the ground than they do in trees, the gibbons being the exception. So there are various stages that we can see in embryological development that reflect the evolutionary origin of the organisms. So Haeckel's work was misinterpreted by saying that embryological development, or ontology, which is the fancy word for this, replays ontogeny, and ontogeny is the fancy word for evolutionary history. That's not what Haeckel actually said, but people have misinterpreted his work to indicate that that is what Haeckel meant. It's not really what he meant. And a close reading of his work indicates that that is the case. What we would prefer to say at this point, and this is the statement that I'm going to put in here, is that ontology reflects ontogeny. Erase that. So what is being implied here is not that embryological development replays the evolutionary origin of us, for instance, because if we were to think about our own evolutionary origin, we would expect that we would pass through a fish stage, which we don't, a reptilian or an amphibian stage, which we don't, a reptilian stage, which we don't, a primate stage, which we don't, but we do pass through evolutionary or developmental stages that where we see features that are related to that evolutionary origin. So the gill slits reflect the fact that, yes, like our very distant ancestors were found in water and did evolve in water, and gills were very useful there, but they're not useful to us now, so we don't need gills. So we don't have gills, but we still have the precursor tissue that would evolve into gills, but that has been redirected to other purposes, as I laid out. And the same is true for other features that we could talk about along the way, and we will talk about along the way as we progress through the semester. So it is not correct to say that ontology replays ontogeny. It is more correct to say that ontology reflects ontogeny. Then the presence of vestigial structures. I've already talked about our appendix. That's a vestigial structure. We see vestigial bones in the constrictor snakes. So the boa constrictors, those are the most primitive of the snakes. The vipers or the fanged snakes are more advanced. They don't use constriction. They use injected venom via the fangs. But the primitive snakes, the ones that are most closely related to the common ancestor of all snakes, which was a lizard that shed its limbs, still have the vestigial bones for the hind limbs. They're not connected to the vertebral column. They're not connected to a pelvis. There is no pelvic remnant, but the little bones are there. This is the same situation in whales. Many whales have these vestigial limb bones. And you also see remnants of vestigial limb structures in things like manatees and dolphins, which are the dolphins are whales. They're members of the cetaceans, but manatees belong to a different group. But you see the same thing there. So where you see this loss of hind limbs as an adaptation to life in water, or loss of hind limbs because of a different means of locomotion, in some of the what we refer to as basal groups, those that are most close closely related to the common ancestor, you do see these vestigial structures. And that is, again, a link to natural selection and descent with modification. Then not only do we see physical homologies, now with our ability to sequence DNA, we see genetic homologies. We see genetic homologies both at the protein level and also at the DNA level. And this is something that we're going to address again in a later chapter, because we can actually use our ability to compare sequence data, particularly at the DNA level, to now establish just how closely related organisms are to each other in taxonomic trees. And a lot of the modern taxonomy, what is referred to as cladistics and cladistic analysis, relies on comparisons of gene sequences, not comparisons of physical features. Because the thing that we can state now, and the thing that hopefully you realize, is that any changes that have led to the emergence of new species or the adaptation of new species must be reflected in the presence of mutations in the DNA. And now that we can actually fairly easily sequence DNA and do DNA sequence comparisons, we can now start to identify at the single base level changes that have occurred in DNA sequences as a result of adaptation, as a result of speciation, the emergence of changes within species, and the emergence of new species. Written in the DNA is the history of evolution, the history of adaptation and descent with modification. So we make a really big deal out of these gene sequences, and we place a lot of emphasis on those now. Now, those are considered to be diagnostic in terms of establishing relatedness, and you'll learn more about that again in a later chapter. So here is a cladistic tree. And I'm not going to talk about it in detail because we're going to revisit this tree in another chapter, and I'll talk about what these branch points represent and how we create these branch points. But this is a tree that shows the relatedness between vertebrates. And you might question some of this. It's not important that you understand the origin of this tree right now, but we will discuss this tree in more detail in another chapter. This shows the relatedness of vertebrates. What I want to point out to you is that when we do sequence analysis of both proteins that are found in common amongst these species, and we do DNA analysis of genes that are found in common amongst these species, they support this particular tree diagram. And this is something that has been very important over the last few years, because we've been able to draw these sorts of tree diagrams on the basis of physical comparisons for a long time. This is basically where Linnaeus started. Remember, Darwin took Linnaeus's tree diagrams and said those tree diagrams exist because of common evolutionary origin, without any particular evidence other than shared common features. We can now dig deep into the DNA and the protein sequences and compare those sequences, and particularly at the DNA level, and say, yes, the changes at the DNA level reflect the changes that, the visible changes that led to this tree in the first place, but we can be more definitive about that. And again, we're going to go into a lot more detail about this in another chapter, so I don't want to keep talking about this. I'd like to finish this up. Then one of the things that Darwin was very much familiar with, and Wallace, was the notion of biogeography, the distribution of organisms geographically across the world, and the fact that organisms that live in the same geographic region bear a lot of resemblance to those organisms that are found in the same region in the fossil record, and are very different from organisms that live in similar types of geographic regions, so temperate zones or tropical zones or things like that, each region, each geographic region has its own unique group of organisms. But at times, we also see that there are similar features amongst organisms living in very different geographic regions. So here's an example. The sugar glider is what is known as a marsupial. It has a pouch, the marsupium, the female does, and the marsupials, as you hopefully know, are the dominant vertebrate life forms in Australia, my home country. These are all mammals, but they are considered to be fairly primitive mammals compared to what are known as eutherian mammals, or mammals that retain the embryos internally in a uterus, and the vast majority of the embryological development occurs within the uterus, and the young are born very well developed compared to marsupials. Marsupial young are born very poorly developed, and they crawl up into the marsupium where the large majority of the embryological development takes place. So here you've got two different types of mammals. Marsupials and placental or eutherian. Now, as mammals, they share a common mammalian ancestor, but the direct ancestor of the sugar glider is a marsupial and the flying squirrel is a eutherian mammal are much younger, lie closer to the present than the common ancestor that they both share as a mammal. So in fact, marsupials as mammals branched off the lineage of mammals that would eventually become the eutherian mammals very, very early and went their own separate way. And the ancestors of all the marsupials in Australia have been completely isolated from all other mammals for about the last 65 million years, which is the reason why marsupials dominate the landscape in Australia, because they had the continent to themselves and they have evolved to occupy all of the niches within Australia, so that when eutherian mammals did eventually make it down to Australia, they found no vacant ecological niche for them to occupy. So they never were able to set up a presence in Australia. In fact, eutherian mammals never really came to successfully settle in Australia until the ancestors of modern Aboriginals brought them in the form of the wild dogs that would become the dingoes in Australia. And then eventually Europeans would bring rats and mice and cats and rabbits and other eutherian mammals that would establish in Australia. But those were the product of man's intervention, not the product of natural migrations. The ancestors of the flying squirrel, however, evolved in a northern large continent called Laurasia, which consisted of North America, Europe, and Asia. Australia was part of a larger southern continental mass called Gondwana land that consisted of largely Australia, Antarctica, and South America. So South America and North America were in separate continental land masses when the ancestors of marsupials evolved versus the ancestors of modern mammals. So these do not share a common ancestry at all, only in very, very distant past. So we can't consider their common features, and you can see that they do share common features, as being the product of common ancestry. So these are not homologous structures. These are, in fact, analogous structures. So the common features here sorry, my writing's a little off there. It's running out of space. So the reason why the sugar glider and the flying squirrel have common features is because they independently arrived at this solution, this same solution to the common problem of how do you fly between trees if you don't have wings. They use instead elongated skin pouches stretching between the forelimbs and the hind limbs. They jump out of the tree and spread their legs out, and they glide between the trees. Where we think this originates from is the fact that there are apparently a limited number of solutions to some problems in terms of behavior in the natural world. So in the case of flight, you either have wings, so you have the bird's wing, you have the bat's wing, you have the insect's wing. They're all analogous structures, they're not homologous structures, but they are solutions to flight in the form of wings. In the case of gliding vertebrates, well, the flying squirrel and the sugar glider have come up with these elongated skin membranes. There are gliding frogs or flying frogs, and there are flying snakes, and they use a similar kind of idea. They have elongated skin membranes that allow them to glide. They don't actually fly. So in the case of flight, you have wings. In the case of gliding, you have this sort of thing. So these are all examples of convergent evolution. We'll come back and talk about convergent evolution again in another chapter because we've got more to talk about in this case. And so when we think in terms of island chains like the Galapagos, and this is something that Darwin eventually came to realize with the case of the finches and the tortoises and so forth, all the organisms that live on the Galapagos must have come from somewhere else at some point, because the Galapagos Islands as a group are only about 3.5 million years old. And logically, it makes sense that they all came from South America, which is only about 600 miles away over the ocean. And so when you look at island chains around the world, what you recognize is that the species that live there are most likely derived from the nearest large landmass, and that's what you find. So when Wallace was looking at the species on the Indonesian archipelago, until he got down to the region that we now recognize as the Wallace Line, everything had migrated from South America, from Southeast Asia down the archipelago. When he got to the Wallace Line and crossed over that, he found that the species had been migrating from Australia or out of the Pacific, from New Guinea and Australia and out of the Pacific and up into the islands, and more reflected that origin. And so we can recognize the origin of the species, but what we also need to recognize is that once they reach those islands, they take on their own unique form as they adapt to the local environment as they find it. The most impressive example of all of this is Hawaii, because Hawaii is about 2,500 miles from everywhere. And so all of the species that are in Hawaii now have come from somewhere else, and typically they've come over long distances, and they've come from different locations around the Pacific. So fruit flies have come out of Southeast Asia, and we can see the progressive movement of fruit flies through the islands. And you can see that the islands are progressively younger as you move eastwards, and that's because the Hawaiian Islands are the product of a hotspot, a geothermally active region on the floor of the ocean, of the Pacific Ocean. But because the floor of the Pacific Ocean is moving over that hotspot, the hotspot remains stationary, but the floor of the ocean is not. As it migrates, depending on the rate of its migration and how long it sits over the hotspot, you get islands forming. And so, in fact, you can track the Hawaiian Island archipelago all the way over to almost Russia. So Midway Island and some of the other islands and what are referred to as seamounts, islands that never quite made it to the surface, form a chain that reaches all the way almost over to Russia, which tells you that this hotspot has been around for a long time. And in fact, another island in the chain is in the process of emerging, and it's emerging over here to the east of the Big Island. It's probably several million years yet before it emerges, but we know that it's emerging because submersibles have been down and you can see the ongoing volcanic activity that's producing it. So the Hawaiian Islands have shown this progressive population by organisms as the islands have emerged. What's impressive about the fruit flies is that there are nearly 500 species of fruit flies endemic to the Hawaiian Islands because of the multitude of different habitats that are present, because the islands are relatively young and they're still geologically active. And so you see many environments for the fruit flies to occupy. Whereas if you go to Southeast Asia, where the fruit flies emerged from, there's only 5 or 6 species, because the continental landmass is a lot older and it is more contiguous or continuous. So there are fewer opportunities for the fruit flies to evolve new species. So it's kind of cool to see that. And in fact, Hawaii is considered to be a major laboratory for evolution. But unfortunately, human intervention in Hawaii has also created circumstances where a lot of the very unique organisms that live in Hawaii are endangered, and a number of them have already become extinct since humans reached the islands probably around 1000 CE or AD, if you use that designation, in the form of the Polynesian settlers coming out of Tahiti that settled this region and brought with them animals and plants that they established on the islands. And from that point onwards, the native animals in Hawaii have always been under threat, and some of them have gone extinct, unfortunately. So what tells us about these movements, and I just noticed that there's a typo here, that should be current, is the nature of the movement of the continental landmasses, what is referred to as plate tectonics. So here you can see some of the movements. We'll talk about this in a lot more detail when we talk about the history of life on Earth, because we'll get into the plate tectonic story there. So I'm not going to talk about this in more detail, but you can see the two large land masses that I was talking about with regards to the sugar glider and the flying squirrel. I had a brain lapse at that for a moment. The presence of Pangea or Pangea explains why the rise of reptiles, and particularly dinosaurs, was favored at around this time, and the reason why amphibians failed to thrive. They had been the dominant animal, terrestrial animal form until Pangea formed. The distribution of animals and organisms in general across the Earth's land masses can be explained by the movement of the continental land masses. The reason why some fossils are found on either side of the Atlantic Ocean that are fossils of freshwater organisms. It's the same organism on both sides of the Atlantic Ocean, but they're separated now by several thousand miles of salt water. The explanation is because at one point North America and Europe were part of the same continent. And it was during this period that those animals were present in the freshwater streams in this large supercontinent. There's a lot that we can explain about the evolution and the history of life on the planet by understanding this simple movement of the continents over time. And as I said, we'll get into this in more detail in another chapter. And then, of course, we've got the fossil record, which is the best evidence of evolution that we have on the planet, for the simple reason that this is the record of organisms that have lived in the past. That's what the fossils represent. That's what Cuvier first recognized. And it does show us that this pattern has been in the form of simple to more complex life forms, something that we would expect on the basis of what the theory of evolution as proposed by Darwin and Wallace both suggested, that we would go from simple organisms to more complex organisms. And if you move back in time, eventually you must get to the original cellular origin of life, something that we'll talk about in another chapter. It is the fossil record, even though it is very, very incomplete, that is the most important historic record of life on Earth, because the one thing that you have to remember in all of this is that 99% of all the organisms that have ever lived on the surface on the planet Earth are dead and only found, if they're found, in the fossil record. Of all the organisms that have lived on the planet, only 1%, probably even less than 1% of those organisms are still alive today, or are alive today. 99% of all the organisms that have ever lived on our planet are dead. And if we have fossils of them, that's the evidence of their existence. And of course, we don't have fossil records of all the organisms on the planet that have ever lived. We have a very small representative sample of what has lived, but it's enough that we can tell the story of life in some detail. And again, we'll talk more about this when we get to the chapter on the history of life. So here's an example of a series of fossils of an early ancestor of arthropods called the trilobite. Trilobites show very little change over time, but the area where they do show change is around what is known as the cephalothorax, the head region, if you want to call it that. It's not purely a head, but we call it the cephalothorax. These are found in layers of sediment, in this case, in the area of San Bernardino in Southern California. I used to actually live in San Bernardino when I first arrived in America. Worked at a university in that local area. And so these are fossils that are found of trilobites that are found in varying depths. So the most recent fossil is found in the upper layer. The older fossils are found in the lower layers. So when we look at the fossils in the lower layers representing the older examples of the trilobites, they are more simple in form than the trilobites that are found in the upper layers, which are the more recent, the younger organisms that show a greater degree of complexity, particularly in that cephalothorax region, where we see most of the changes present in trilobites. Not that they show a whole lot in the first place. Sometimes we can see evidence in the fossil record that shows relationships that we might consider to be really unusual, but today, with our DNA evidence, we can actually demonstrate that the fossil evidence is absolutely correct. So when we take a look at the ankle bones of what are known as artiodactyls, these are even-toed ungulates. So ungulates are animals that are herbivorous, and in the case of some of them, they chew their cud. In other words, they regurgitate their food and chew it a second time. And they have a very unique ankle structure with two lobes in a very specific particular location on the base of the ankle bone. So pigs belong to this group, cattle belong to this group, cameloids, so the camels, the llamas and so forth belong to this group, the deer and the antelope belong to this group. If you look at most other mammals, they have a single process on the base of the ankle bone. When we look at whales and when we look at the fossils of the ancestors of whales, they very uniquely have an ankle bone with the two processes on the base, like the pigs and the deer and the cattle and the hippopotamus and so forth. So the artiodactyls and the cetaceans share common ancestry, or more specifically, the whales share a common ancestor that was part of the artiodactyl group. And that's seen here in the structure of the ankle bone. And this is true also in the ankle bone that's part of the foreflipper of the whale that we were talking about earlier. We see this, the ankle bone in modern whales that possesses these two processes in this location on the bottom of the ankle bone. At the DNA level, we can see this common ancestry now, because we see DNA sequences that are reflective of a terrestrial ancestry. But those genes are now what we refer to as pseudogenes. They've been disabled. They no longer function as genes, because they are genes for things like being able to smell chemicals in the air. Well, you obviously can't do that if you live in water. Whales use a different way of detecting chemicals in the water, but they still have all the genetic sequences. They're non-functional genes now, pseudogenes, but they show that the relationship between whales and their terrestrial ancestors is a very ancient one, and that in the case of whales, the genes that no longer served a purpose became non-functional. Remember the cost-benefit analysis? If it costs more to maintain something than the benefit that you get from it, and here's something that you get no benefit from in the water, then you lose it. And there are many examples that I could cite for you, but the whale one is one that I've spent a little bit of time investigating, because I have used this in a seminar that I gave many years ago here at Valencia about evidence for evolution. And so this one's one that's is one that I know a little bit about. So we see all of this evidence present in the fossil record, and it gives us confidence that the way in which we understand evolution to work, simple to complex, descent with modification, tinkering with existing structures rather than creating new structures, all of this is identified both in the fossil record and is strongly supported now by the DNA evidence. So this shows you the origin of whales and the connection back to their closest living relatives, but also to the ancestors that we have in the fossil record. And Pachycetus, which was found in, of all places, Pakistan, is actually the terrestrial tetrapod ancestor of all modern whales, based on the ankle bone evidence and some other evidence that we have. We might get into a little bit more detail in a later chapter. So is there anything theoretical about Darwin's view of life? Well, it depends on how you want to use the term theory. If you're using the term theory as it is used in everyday language, then the answer is no. There is nothing theoretical about the theory of evolution. If you are using the term theory as it is defined in science, and if you look up the term theory in a dictionary, you'll find both definitions, then the term theory in science is very much appropriate, an appropriate term for evolution, because it is an overarching explanation of a whole lot of observations, which is what we define a theory to be. It is a predictive model, meaning that if you take what is stated in the theory and make predictions on the basis of what the theory says you ought to be able to find, when you go out and do the experimentation or do the studies, make the observations, you find that the data that you collect is supportive of that predictive model. And of all the theories in science, evolution is the one that has been tested the most by making predictions, and under all circumstances where the predictions have been put to the test, the theory has stood that test on the basis of the data that has been collected in support of the prediction. Of all the theories in science, the Darwinian theory of evolution, or the Darwin-Wallace theory of evolution, is the one that has the most experimental support. And this is where the work of the Grants, Peter and Rosemary Grant, comes into play. And I would strongly encourage you to go and take a look at the videos that are available in the study area of mastering biology, because this is something that you may not get a chance to look at in many other places, because the Grants have made a contribution to Pearson in the form of these videos so that students can learn about the work that they've carried out in the Galapagos and understand the significance of what they've been able to do, because they have not only identified changes in beak size as a response to environmental changes on the island that they've been working on in the Galapagos, but they've also identified the emergence of a new species during that time. Over about the last 15, maybe 20 years, they've been able to document the emergence of a new species of finch on the island that they've been working on. So here is a situation where speciation has been observable during the course of a human lifespan, something that we never thought we would be able to do. Because we've always thought that speciation was a long, drawn-out process taking hundreds of thousands of years, maybe millions of years, for the small changes to accumulate to give rise to a new species. But the Grants have been able to demonstrate, and a number of other situations have been able to demonstrate as well, that speciation may take place a lot more rapidly than we may have been able to imagine based on our understanding of the theory of evolution. Speciation may actually be a very rapid event, both with respect to geological time, and we'll talk more about that in another chapter, but also in real time. It may take place a lot more rapidly than we previously thought. So one more concept check, and then we're done for this chapter. So how does evolution account for the similarity of mammalian forelimbs and the similar forms of distantly related mammals? So this time it's not a break. This is the end of the presentation once we've got this question answered. So remember, mammalian forelimbs are similar because of descent with modification. They share a common descent from a common ancestor. So the forelimbs are similar because they are modifications of that original ancestral limb structure. So that's how we explain the similarity, and it's modifications, descent with modification, for use in different ways by the different types of animals, different behavior, different environments. The similar forms of distantly related mammals is this notion of convergent evolution. And that's exemplified by the sugar glider and the flying squirrel, but there are any number of examples of convergent evolution that we could propose. The bird's wing versus the bat's wing, those are analogous, an answer to the problem of flight, real flight, by developing two different types of wings independently, one in mammals, one in dinosaurs, because birds are dinosaurs. Yeah, you heard it first here. Birds are truly dinosaurs in every sense of the word. And I heard just a few days ago several paleontologists and biologists reiterate that very, very firmly and very, very confidently, that dinosaurs are still living. You may very well be eating dinosaur tonight for dinner, if you have chicken. Anyway, the reason why these similar forms exist between very distantly related organisms is because of convergent evolution. Converging on a common solution or a similar solution to a common problem. In this case, with birds and bats, how to fly using wings, and then insects come up with a completely different version of the wing. And then in the case of the sugar glider and the flying squirrel, how to glide between trees without wings. And they arrived at the similar solution of having skin stretched between the hind limbs, forming a gliding quote unquote wing in a similar form. Think of a paraglider or something like that, or even a modern parachute is more of a gliding wing than anything else. So they came up with that solution. So that's it for this chapter. It's I realize there's a lot of information, and this is, if you were to put all of these recordings back to back, it would be quite long, but you can break them up at the points where I've broken them up and just focus on each of those recordings one at a time. And hopefully this will provide you with enough information and explanation so that the chapter is easy for you to understand. It's not a difficult chapter, but hopefully I've given you a little bit more information that make it a little bit easier for you to understand. As always, if you have any questions, concerns, or comments about anything that I've addressed in these presentations, by all means, come and see me during office hours or email me, whichever works for you. So until next time for chapter 23, be safe, and I'll see you then. Bye.